Hi. So far we discussed in situ and ex situ biodiversity conservation method. In all of these methods, the organisms are conserved in a living, active state. But the advancement of technology leads to the methods in which we can actually store the organism in a such a way that it will be stored for a very long time. In the following sessions, I am going to talk about this type of ex situ conservation method. We can begin with the gene bank. Gene bank conserves genetic resources. As the name implies, it is a method of conserving genetic resources. It stores, maintain, and reproduce living samples. So it is actually a storage, not like a botanical garden or the national park. It's actually we are storing something, and if we need it afterwards, we can actually regenerate the whole organism by this stored commodity. It's a biorepository which preserves genetic material for long term. We can do it for plants by seed banks, in vitro storage, or cryopreservation. For animals, we can preserve the sperm or egg of the animal, and from that we can actually regenerate the animal if we need it in some time by using in vitro fertilization. Or we can preserve tissue, chromosomes, or DNA of any plant, animal, or microorganism. We can preserve its tissue using some techniques, or chromosome, or even if we have the DNA, by using the genetic manipulation and genetic engineering, we can reproduce the whole organism. For unlike other conservation techniques, in the gene bank we are conserving organism in a stored manner, not like the living physiologically active state. It is a reduced metabolic state. We don't need to feed the organism. We don't need to do anything. We are conserving maybe it in a refrigerator or in a dedicated space. So conservation is happening like that. So there are some prerequisites for conserving something in gene bank. First thing, understanding genetic diversity of the population, because in order to have a representative gene bank, we should take samples from all variants of the population. And there should be scientific sampling to represent the whole gene pool. It is a method for preservation which prolong viability while ensuring the quality. So the methods of conservation, whether it may be at a low temperature or maybe some other conditions which reduce moisture or reduce pressure, whatever condition, whatever preservation techniques we are using, this technique should be able to prolong the viability of the organism without losing its quality. We need to maintain the controlled conditions during the preservation, which is a very important point. But like if we are conserving the organism, maybe large number of organism in cryopreservation, which means the low temperature storage. But if some failure, some technical failure occurred to the equipment, what happens? The whole organism stored in the equipment will be lost. So controlled conditions during preservation is very important. There should be alternative power supply, maybe alternative equipments, so that the technical errors or things like that, natural disasters, should not affect the preserved organisms. Regular monitoring to ensure viability. The stored commodity should be regularly inspected so that its viability won't be lost. Proven methodology for regeneration. There should be methods to regenerate the preserved commodities into living organism, and it will vary depending on the technique. For one technique we need this type of regeneration strategy, for another technique we need another type of regeneration strategy. And proper characterization and documentation at all levels, because it's a storing, unlike the living organism we are storing it in some refrigerator or something like that. So proper characterization and documentation is primarily important. In all this gene bank, we are preserving under appropriate conditions for increasing the viability for a long period. There are different methods we can use. One is cryopreservation, that means low temperature storage, or we can use artificial ecosystem, which reduce the rate of growth, or control the nutrient medium. We can actually in vitro grow the organism or its cells or components in cell cultures, and we are when we are giving medium which has less nutrient capacity. So the growth will be retarded, but that retardation can be used as a tool for preservation. Or we have some slow growth conservation methods also, which actually reduce the growth rate but maintain its viability. And whatever gene bank storage we are adopting, the frequency of regeneration should be less, because if the frequency of regeneration required is high, the genetic diversity may be lost. The regeneration means if you are storing one plant or one part of the plant which can be used for vegetative propagation in a refrigerator, at regular intervals you need to regenerate. We take out this vegetative part and plant it in the soil and regenerate the plant, and again take the vegetative part and store again. So that's a regeneration process. If you are doing it so frequently, the genetic diversity will be lost and the chance of contamination will be high. So that's not recommended. The ideal preservation method should not have many regenerations. The frequency of regeneration should be less. And this stored commodity should be available for researchers and breeders for scientific and industrial purposes. One type of gene bank is seed bank. Actually, seed bank is a type of gene bank. It's a type of gene bank. It stores seeds or embryos to preserve its genetic diversity. So, by just storing the seed, we can actually store the organism. But not all plants will make seeds. If it is making seeds, we can actually store. We have traditional method of storing these seeds, but these methods will be able to store the seeds maybe for a few years maximum. But right now we have the advanced methods and techniques in which we can actually store the seeds for thousands of years. So seed bank, it is the most preferred mode of conservation for plants because seeds are convenient and easy to collect. Then it occupy a very less space. It maintain viable state for long periods. Naturally, it has a long viability period. And it is most cost effective method because collection, conservation and regeneration, everything is easy. Endangered plants can be conserved by using this method. We can actually easily regenerate these plants. It can be used for crop varieties as well as for the wild varieties of plants. And after the storage, it can be made available for farmers, breeders and researchers for purposes. And this seed bank can be used for short term, medium term and long term storage. So it is very flexible method. And there are some recommended standards for seed banks. It is not just storing the seed, it is a scientific method of storing, preserving the biodiversity of wild and cultivated varieties of plants. So International Plant Genetic Resources Institution and the Food and Agriculture Organization has developed some standards in which a seed bank should proceed. For a proper seed bank, there should be large populations of seeds should be preserved. There is no few number of seeds, large number of seeds, so that it will represent the genetic diversity. Genetic erosion should be minimized. Reduce the need of regeneration. As I said before, if you are regenerating again and again, which leads to the loss of genetic diversity. Cryogenic storage is the most preferred method of storage, the low temperature storage, because it can conserve the seeds for centuries. Care should be taken during seed collection, regeneration and storage to avoid contamination. Continuous viability monitoring and regeneration and recollection should be done. Recollection means that at regular intervals we will collect the samples from the wild or cultivated plants and store. So regular intervals we are actually supplementing the storage by new entries. So that's what is called recollection. Documentation at each stage of activity. These are the recommended standards for seed bank. The seed banks can be stored in different ways. Different methods can be used for preservation in a seed bank. Storage of seeds are usually done under controlled conditions, which means reduced temperature, reduced seed moisture content. If the seeds are dried, in most of the cases if the seeds are dried, the life will be extended, but some cases it doesn't work. And controlled relative humidity, that is the humidity of the place where it is stored. If it has high humidity, as we know, some seeds may lose its viability or it will start to germinate. So that should be avoided. That can be avoided by controlling the relative humidity of stored place. For long-term storage, what is the recommendation is that seed should be dried below 7% of moisture, and after drying, seeds are sealed in a moisture-proof container made of laminated foil bags or aluminum cans or glass jars, and these jars will be then stored in low temperature, maybe minus 18, minus 20, minus 22 type of temperature. And preservation methods is always depending on the type of seed and behavior of the seed. Some seeds are called orthodox seeds. The thing is that the orthodox seeds are desiccation tolerant. If you dry the seeds, the seeds can be dried and stored for a long time. Those type of seeds are called orthodox seeds. And the other type is recalcitrant seeds. They are desiccation intolerant. If you dry the seeds, its germination capacity will be lost. So such type of seeds are called recalcitrant seeds. So the behavior of the seeds is important when we are making a seed bank. We cannot actually dry the recalcitrant seeds. We can only dry the orthodox seeds. Then quantity of seed production. If the plants are producing huge number of seeds, it is easy to conserve. And duration of life cycle. If the plant is having a very large life cycle, it is impossible to store the plant in the seed bank because we need regular regeneration. If you want a regeneration after every 5 years, if you are storing a plant with a life cycle of 100 years, it will be very difficult. After 5 years, you need to germinate the plant and wait for another 20 years to get the seeds. Then only you will be able to regenerate, restore. So that's impractical in most of the cases. These are the steps of seed bank operation. First one is collection. There should be scientific collection methods which helps to maintain the genetic diversity. Then seed preparation. Prepare the seed in such a way that you're cleaning the seeds and everything so that we can preserve. Sometimes the microorganism should be killed from the surface or removed from the surface of the seeds. Then seed drying, which is a common practice for most of the seeds because drying helps it to live longer. Then packaging to avoid to keep the relative humidity checked. The packaging is essential. Then storage will be in reduced temperature in most of the cases. Depending on the behavior of the seed, we will select the temperature. Then once it is stored, there should be periodic germination tests after regular intervals, maybe 5 years or 10 years. We need to take few of these seeds and try to germinate. If it germinate, everything is fine. But if it does fail to germinate, we should recollect the seeds or trying to regenerate most of the samples. So the thing is that when we are storing the plants, we should do in large numbers. Then only we can take few seeds and germinate. Otherwise, we will lose all of our seeds. Then seed regeneration. Whenever we need the plant back, we take the stored plants and regeneration. And from the regenerative plants, few of them will go to the industry or the research or for the farmers, and rest of them will make seeds for re-storage. That way we can actually keep the storage for a long time. There are some limitations for seed bank. One thing is that loss of viability over passage of time. If you are storing for a very long time, it may lose the viability, and if you fail to notice the loss of viability, you will lose the plant. Then susceptibility to insect and pathogen attack. Some cases we are not storing the seeds in refrigerator or such equipments. In those cases, the insect or pathogens can come and attack the seeds and reduce its viability. Then inability to maintain distinct clones. Then non-applicability to vegetatively propagated crops and perennial plants. The vegetatively propagated plants don't make seeds, so you cannot store it in the seed bank. They have no seeds. Okay. The perennial plants will make seeds, but they have long life cycle. They live more than 3 or 5 years. Maybe some of them will live for centuries. So they will take long time to make seeds. So this seed bank is not practical for them. Then natural selection cannot be stimulated because it is sitting in a refrigerator. There is no way of natural selection. And there will be some form of artificial selection. Some of the seeds which is resistant to the storage condition will germinate faster and they will have an advantage, and which may have unpredictable genetic changes. One another concept is community gene banks. It's an informal seed bank in developing countries, especially in India, because farmers are using, they are not buying the seeds, so they are not always buying the seed from the market or from industry. What they do, they rely on seed from the previous harvest, and the leftover from the previous harvest will be used as the seed for the next generation. And there are traditional system of storage, treatment, and the exchange of seeds. They are mostly informal in nature, so people will collect the seeds and some people will be regularly making the seeds and they will distribute to other farmers for money. So it's actually the exchange is within and between communities and in an informal way, and there is an indigenous structure for information flow and the exchange of seeds. Whenever they are selling the seeds, they will be describing its properties and how it is should be planted and how it should be taken care of, and then how it should be harvested, what is the period of germination or period of growth, and what all care you should give. So such all things will be communicated between these communities or within these communities in an informal way. So it operates in very small scale at a community level, but for conserving local varieties of plants, the conserving the community seed bank is important, because the local varieties what farmers are actually using in the field is circulating in these communities in an informal way. So these structures, this community gene bank structure, the community exchange of seeds should be conserved so that we can actually conserve the agriculturally important varieties of plants. Thank you so much.